Hi everybody, I am Karno. I'm an associate professor from the Department of Chemistry and Biochemistry from California State University, San Marcos. This project I'm doing with my students, Michael, Nick, Adan, and Victor. So the topic is development and extraction and detection method for a chemotherapeutic drug with phenytoin in a biological sample. Usually when a cancer patient with, especially with brain tumor, they sometimes often have a seizure. These two drugs, dexamethasone and phenytoin, are usually given concurrently to the patients when they first being diagnosed with primary or secondary brain tumors. Phenytoin is administered even when they don't have seizure yet. It's as a prevention treatment. So let's first talk about these two drugs. So dexamethasone was approved by the Federal Drug Association in USA in 1958. It is used for different treatment. It can be used for treating cancer to decrease the inflammation, and sometimes it is being used for before or after the chemotherapy to prevent or treat nausea or vomiting. This is the structure of dexamethasone, and as you can see here, it's being administered in different, either the tablet or injection format. And phenytoin is approved by FDA in 1939 for the treatment of seizures or epilepsy. This is the chemical structure of phenytoin. Okay, so it is, the way it works is it exerts anti-seizure activity, but it will not cause a depression of the central nervous system. The phenytoin has a very narrow theoretical index. By definition, the theoretical index is the range of the dose at which the medication is effective without a very significant side effect. So that's why phenytoin is a drug that has to be administered very carefully because it has such a narrow range. So here is, again, just like the dexamethasone, it's in tablet format or it can be as injection. So the previous study has shown that phenytoin will decrease the level of the dexamethasone in the patients when they are being given concurrently. On the other hand, inconclusive observation has been seen for the effect of dexamethasone on phenytoin, because both the decrease or increase in the level of phenytoin has been observed. So because of this observed the interaction between these two drugs, and these two drugs are being given concurrently to cancer patients, it is very important to monitor both of these drugs concentration in order to make sure that the effective and ideal dose are being given to the cancer patients without any adverse effect. So the way we do our experiment is we first develop the high-performance liquid chromatography conditions. We use a Sumatsu HPLC. It consists of two pumps, and it has the detector we use is a photodiode array. And we detect the dexamethasone at 290 nanometer, and phenytoin at 290 nanometer and dexamethasone at 241 nanometer. A C18 HPLC column is used from Phenomenex, and the amount we inject, the sample we inject is 10 microliter. And then the mobile phase consists of three components. It consists of the 0.001 molar KH2PO4, and then the acetonitrile and methanol, and the whole mobile phase is adjusted to a pH of 5.6. The proportion of this three component is here, 48 to 32 to 20 by volume. The flow rate is 1 ml per minute. So in order to test that our detection is in a good linear range, we first establish the calibration curve. We do that by preparing a stock solutions of phenytoin and dexamethasone, and then we prepare 5 standards, okay, to it, and each standard was injected 3 times, and then the peak area is recorded. So this is the calibration curve for dexamethasone. We plot the concentration, it is microgram per mil, versus the peak area. As you can see here, it's very linear. This is the calibration curve for phenytoin. Again, we are plotting the concentration versus the peak area, and it's very linear. Oh, I'm sorry, should be flipped. The peak area should be here. Yeah. So the preparation of the sample, so we spike a 20 ml of the urine sample because we cannot, I mean, we are not at the medical school, so we can only, we cannot directly give the medication to the patient. So the way we do it, we mimic it. We used, we got 20 ml of the urine and we spike it. That means we're adding both drugs into the urine sample, and then we extract it back out. So as you can see here, we spike it with 2 stock solution. The phenytoin is 563 microgram per mil. The dexamethasone is 578 microgram per mil. We put 1 ml each in here, and then the resulting concentration is 20 times less. So that's 28.15 and 28.9 microgram per mil. And then we vortex this to make sure that it mixes into the urine sample. And a blank sample is being done exactly the same, except, you know, instead of using the urine here, we use distilled water. So after we put the both drugs into the urine sample, now we are extracting it back out. And this is the condition. We are using a 24 parts vacuum manifold from Phenomenex. This is the diagram to show it. So we are using a C18 stranded cartridge from Phenomenex. The cartridge is first conditioned with 3 ml methanol, and then followed with 2 ml of water, and finally we put 2 ml of 0.1 molar phosphate buffer at pH 6. Once we condition the cartridge, then we start loading our urine sample in it. We load it with 3 ml of the spiked sample. Then we wash it with 3 ml of distilled water, and then followed by 2 ml of 20% methanol. Finally, we elute our sample with the drugs in there with 3 ml of methanol. Once we elute the sample, we dry it under nitrogen gas. To dry the sample, then we redissolve it with 1 ml of methanol. And then we filter it with a syringe filter before we inject it to the HPLC. So here's the chromatogram for phenytoin. As you can see here, the retention time is it comes out about 8.3 minutes, and we detect it at 219 nanometer. And then for the dexamethasone, it comes out around 10.6 minutes, and we detect it at 241 nanometer. So then after that, we calculate the percent recovery. The way we calculate it is we compare the peak area of the analyte. For example, if we're analyzing phenytoin, then we find the peak area of the phenytoin in the extracted sample over the peak area in the blank, and we multiply that by 100%. So then we perform interday and intraday study. Here's the result for it. We do 3 days, and then we compare those 3 days, and that's the interday. As you can see here, for both drugs, for both the phenytoin and dexamethasone, the percent recovery is pretty good. For the intraday, for phenytoin is about 93%, range from 92 to 93%. And then for dexamethasone is around 78%. And the relative standard deviation, which is the measures of how close each measurement is very good. It's below 4%. And the interday study, we do it 3 times at 3 different days. It's also very, the percent recovery is very similar to the intraday. and the relative standard deviation is relatively low. Then we try to do a comparison study to spike it at a 5 times lower concentration. So as you can see from this diagram, we're doing exactly the same extraction process, but this time we are spiking our urine sample with a 5 times lower than what we spiked it before. So the resulting spiked sample is 5.6 and 7.78. And here is the results. Again, the percent recovery is very similar to the previous spike level, showing that this method is applicable for different spiking level. So this is just a comparison. So at a original spiking level at about 28.15 microgram for both drug, the phenytoin and dexamethasone, comparing with the 5 times lower spiking level, as you can see, this is the RSD is comparable and the percent recovery is also comparable. So the final conclusion is, as you can see, the calibration curve shows a very good linearity. The R-square value is greater than 0.99. And we also determined the detection limit for both drugs is about 0.0062 and 0.0072 microgram per mil. The retention time for phenytoin and dexamethasone is 8.33 and 10.69 minutes, which shows that we can finish the analysis within 10 minutes. And we already seen the percent recovery for both drugs is very good in two different spike level. For phenytoin is about greater than 93%, for dexamethasone is greater than 78%, and the RSD is smaller than 4%. So the data shows that the developed extraction and detection method is efficient for extraction for both drugs simultaneously from the urine samples. And lastly, this is the list of the references. And thank you, and I will be happy to answer any question if you have any.